CCT is happy to do this for the LSU campus. LBRN covers the rest of the state. So welcome. I know that there are some people who are connected online. So for everyone, let me know if you have any questions. I'll be here. Otherwise, I think it's time to start. Thank you.
Okay.
No, we didn't know you'd be talking, so we didn't have you on.
Hello, can you hear me?
Yes.
Yeah? All right, great. Hello, everyone. My name is Ilya Brodsky. I'll be doing an overview today of what we will try and cover in the next several months. And I want to start off once the lights are figured out. Good? Okay. So the goal of this program is to really bring together people that come from different backgrounds, and I think that's both an opportunity and a challenge. What we've learned from the previous time when we started doing this is that we have to learn a lot about how to adapt the materials to the different types of people that are interested. And so we're hoping that this will be a learning experience for all of us, for you and for us as well, because we would like to see and understand from you more about how to make this more useful and effective for you. To do that, I think we have a great team that wants to kind of facilitate this task. There are some people here today. I don't know, some of you are probably familiar with Dr. Kim, Young Kim, who's here. John, Sahil from our side. So, Lyndon, I think Coghill is in the room somewhere. So it would be good at some point for all of you to be familiar with these people because they're all here to help make this work for you. So let me start by saying that the main reason why this was put together is because there is a growing gap. Okay, so there's Lyndon, who is going to be helping us connect between the LSU campus and the people that are online. Okay, so there's a growing gap between the data that is generated and our ability to process that data. So there's a lot of data that we can generate in the lab, and then it's not so clear, how can we use this effectively and what we can do with that data. And so to bridge that gap, we are trying to look at the different stages where this can play a role, where this can have an impact. And this really spans the life cycle of data. And I think it's critical for everybody who's involved in, specifically as we are part of the LVRM, the Biomedical Research Network program, and we're focused on the biomedical projects. So it's important to kind of see this in the biomedical data life cycle of data, where we have the people that generate the data, and they have this task and this goal of generating more and better data. And then we have people that have to analyze the data, and they have to understand.
You forgot to hit test share, so our 5 people are not seeing, so I have to skip. Right. Back on. Sorry about that. Remote sites should now have a test share.
Okay, so we'll go back a little bit. So what I started saying is that there's a growing gap between the data that is being generated and our ability to effectively use that data. And to close that gap, it's important to involve everybody in the process of learning about the data, about its significance, about how it's generated, about how it's used. And that spans the life cycle of specifically biomedical research data, right? So there are people that generate the data that have the goal of generating more and better data, because that's critical for our ability to understand what's going on. The people that analyze and integrate that data, they have to be able to understand where it's coming from and where it's going to design better methods and to improve their ability to integrate the different data sets. And then eventually this data is used in critical decisions. And this is critical for research, for our basic understanding of what's going on, as well as translational applications, how it's used for clinical decisions and for other decisions that impact how we use the data. So it's important for us to connect the dots, to make this a clear process, and for everybody to be informed and able to practically understand and appreciate what happens with that data and where it comes from and where it's going. So to do that, we've put together this program, and I should say that the program is still in development because we're learning, as I mentioned in the beginning, how to put together the different pieces. But what I want to do today is share with you kind of where we are with our understanding of how to make this effective. And hopefully throughout this 6 months, we will be able to identify the missing pieces and hear back feedback from you on how to make this better. But today, just to give you an overview of what this includes. So the idea, the kind of vision for this actually started at the Tauber Bioinformatics Research Center, which is a bioinformatics research center at University of Haifa in Israel. They created a functional platform with the vision of giving people easy to use access to analytical tools for all kinds of data. And the idea was to make processing of that data, analysis, and integration an easy task so that anybody could do it. What we eventually realized is that easy to use tools is not enough. You really have to supplement that by 2 more things. And the 2 other things were tutorials or modules. We call them modules that talk about the different types of data, and projects. Because in every case, you have to have an example project to really understand what's going on and how a data set is formed. So as a result, we have a collection of tools or of sites that we will look at today and try to implement in this program. So the educational modules that came out of this platform consist of these different modules that explain the logic of the analysis. So we call them omics logic because they really take different omics data types and they explain the logic of what to do with them, where they come from, how to analyze them, how to interpret them. And they are kind of centered around your personal progress. So again, the reason they are modules is because this is not a standard textbook class where everybody has to do the same thing. It's really up to you which modules are useful for you or interesting to you. And so hopefully you'll be able to pick and choose from the menu and create your own program as we move along. So because of that, or to finish off that, in every module, there will be a number of projects that you can also select from. These are going to be on various topics and with various types of data. These are just some examples. As I mentioned before, this program is going to be a little bit more on the biomedical side, even though I imagine that there are some people involved in the program that have a different focus. But we will use examples from the biomedical field, and a lot of them are going to be in oncology. So they're all taken from publications, and we kind of digest little sections of the data sets that are published, translate them into small exercises. And then the reason why we feel that's a good approach is because you can then take the rest of the data set, or you can take another part of the data set and make it your own. So it's a known fact that publications only report, a small percentage of what was in the data. And so therefore, there's probably, 90 to 95% of data that you can still do something of your own with. And I think also a lot of them, and this is a good example, this publication here, they actually themselves rely on other published data sets, right? So it's how you collect the data and what you can do with it and what's your focus that makes it your own. So our hope is that at the end of this, you will be able to do what some of the other participants did last spring and in the summer, where we ran similar pilots. This was a pilot at University of Loyola. And then in the summer, of course, we did the LBRN training, where groups of students were able to take some of these data sets and come up with their own ideas and then share that with their mentors and kind of guide it to an interesting direction. So our hope is that throughout this course, some of you will come up with these project ideas, and hopefully during the annual Bioinformatics conference, some of you will be able to share what came out of this course, hopefully with public or your own data set. So the idea is to really go through the logic of the analysis, provide you with the tools, provide you with some example data sets to kind of get the process started. And then as we envision the future of this program, maybe some of you will decide to actually use these resources, develop your own projects out of this, and then we will see more data-driven projects in different research areas. So what we kind of see as a part of this program is providing access to tools and enabling infrastructure. And I think this covers not just what we do here, but in general, maybe that's the objective of the Louisiana Biomedical Research Network, is that this is not just a course to kind of complete it, get a certificate or credit for. It's really to introduce you to these research infrastructural solutions that are available. Exposure to kind of how is this done in different examples, and in different data sets, and then provide an opportunity to kind of take that into your own direction and take it into a variety of projects that either we curate and we put up as an opportunity or start with your own. So the goal of the program is to introduce people that are interested to the resources available through the LBRN, to provide access to tools and methods for data analysis, to help develop independent research projects, to leverage public domain data sets, and then to facilitate meaningful collaborations. And by meaningful collaborations, I think what we've seen in the past, in the summer, for example, is that we will have students or faculty with different backgrounds in biology, and then others with computer science and more technical skills. And I think the real exciting opportunity is when those join forces and they decide to work on a single project, and that project gets farther along because now people can speak the same language, because it's clear what the biologist is trying to do, and it's clear why somebody with computer science background could be really helpful to take the project farther. But it doesn't happen, right away. I think there's a big barrier here to cross, and we hope that some of the material that we prepare will help us to establish kind of like a framework, and then throughout these breakout sessions or workshops that we will organize, we will try and work through some examples and learn about how these different components fit together. Okay, so what are the specific resources that we will make available throughout the program? The first one is user-friendly tools and methods. So by user-friendly, this is a screenshot. I will give a short demonstration of what the platform really does. But it's really a graphic user interface for public domain tools that you can download from GitHub and other places, and you can essentially run wherever you want. Right now, throughout the program, you will be able to use our platform that is kind of centralized, and we can help monitor what's going on behind the scenes so that if there's a problem, we can make the problem go away or at least address the problem. And we can also see how people are interacting and using different tools, which gives us a sense of who is practically doing things and who is involved in maybe just a more theoretical way. The second is online training modules. So another challenge, as you can imagine, is that people are in different locations, different schedules, and so we want to make materials available online, and we want to make these kinds of sessions possible as well. So what we will do is we will have a series of online lectures or modules, and I think that it's very important to try and at least look at them so that you're familiar with those concepts before coming to the workshops. Then, as I mentioned, these will be project examples, and project examples are really data sets with an explanation, typically a publication, that explains where the data came from, what they did to the data, and what was their interpretation of the results. And we will try to replicate every stage of that process and at least give you an idea of if you were doing the exact same thing, this is how you would do each one of the steps. And then obviously you can take it into different directions. And then we have created this kind of a community where we brought everybody together so that you can know who else is a part of the program. So hopefully you will see other people that you could work on different projects together eventually, and learn from each other from different backgrounds and different people that are involved. Now, the other resources that we will touch on or utilize somewhat are going to be RStudio. So we're not going to go deep into RStudio or really learn how to write scripts in R, but we will touch on that, and we will show you how to do simple things like visualization. We will, of course, use Excel because I think we found that the majority of the people already know Excel well enough so that they can use it. But you're welcome to use any other scripting, visualization, or data management tools that you want. Really what we want to provide is just a framework of how to get started. The other part of it is open public domain data sets on NCBI that we will take raw data from, and we will try and learn how to process that data. But it's important to note that all of you will have educational accounts which limit your amount of data that you can upload. So because we are supporting about 100 licenses here, what we've done is we've preloaded a number of resources on the platform, and everybody can have access to those through reference links. So we will show you how to download and upload those files onto the platform. But if you would like to do that, you would have to come to us and ask to open up your limit, and then you'll be able to do that. So just keep that in mind. It's possible, but you do have to kind of get permission to do that. And then finally, we'll talk about reference databases where we can validate some of our findings, right? So if we found some interesting pathways or whatever, and we want to validate in other data sets, we'll talk about how to do that, how to research other databases for that. So one of the things that we will talk about is how the data gets assembled and how to create a project that will have enough samples, and we'll have enough statistical significance for us to kind of base our hypothesis on. And so we will do some exploration of data sets available on NCBI so that you can start thinking about your independent projects. If you do have your own data that you would like to analyze, I know that I've heard from some people that they are already thinking that they could kind of do that throughout this course. Like I said, it's possible. Let's kind of try and coordinate that so that we're aware of what exactly you're trying to do and how this could be useful to the rest of the people that are participating in the project. Okay, so let me demonstrate to you. I think all of you should have gotten an email with credentials for the platform. If you brought your computers, maybe we can just make sure that it works. And I will show you how to log in and how to kind of navigate the platform so that you're aware of what's there. And that's another thing about these kinds of meetings and workshops. Obviously, this is the first one, and so we're just kind of learning what this will be like. But in the future, if we do an in-person meeting like this, please come with your laptops and ready to, have the data set ready, have whatever we set you ready. And if you don't have something working or you don't have some links, just there are some key people that are here, like Lyndon, I think, that could probably help, or Sahil, who's in the back here, but he's also on chat. So let me walk you through some of the links that you should have received by email so that everybody knows what we're talking about exactly. Okay, so the first link that you should have gotten is this link, server.t-bio.info. I don't know if you can see, it's right there on top. And what it is this platform called TBioInfo. This is the areas of analysis. You should have all gotten an email with your login information. So as you can see, this is my profile. So all of you have an educational profile on the platform. Again, the link is server.t-bio.info. Now, if you don't have your credentials with you today, I'm just going to make this visible on the screen. Okay. So I know that some of you don't have laptops. Sorry, probably should have told everybody that it would be useful to have them with you today, but we're not going to actually do anything practical. I'm just gonna show you. Go ahead.
I have received the links because Friday.
Yeah.
So I appreciate it. I don't know what I should talk to get these links.
So everybody that was registered got an email. If you didn't get an email, then please write us so we know who to send it to.
I got the email, but I didn't see the.
You didn't see the link?
Yeah.
Okay. Well, let's just make sure, so we know who to send it to. Did anybody else not get the links? Okay, so 2 people. Okay, yeah, so that's good. So let's try and figure that out today. I mean, we'll have some time today, so come talk to me, or like I said, Lyndon, Sahil there in the back can definitely. Sahil was the one that created all the accounts. So if you have any problems with that, he can help with that. And the other thing is that all of this is really on this educational platform. And let me, okay, so there's an organization page. Let me maybe start from here then. So this link is edu. Okay, so I'll just copy this here real quick. So these are the 2 primary links for us to start with. The edu site is where the educational content is. The server site is where the practical methods are. So please make sure that you have accounts on both. Just a note about the accounts. There are going to be there's going to be a login. So you use your email and you have a password for both of those sites, but you should have gotten 2 different emails about it. Okay? Okay, so on the EDU site, there's a whole page dedicated to this organization called LBRN. So this will be Organizations Louisiana Biomedical Research Network, LBRN. And here you have most of the information that you will need. So you will see announcements here about important events and assignments and kind of what are we doing this week or this month. You can also see an overview of the curriculum that will show you month by month what we are doing and where we are. So today we're doing Introduction to Bioinformatics, and then next week we'll be doing Introduction to RNA-seq, and these are going to be the online modules that you can look through. Then you will see in December we will do epigenetics, and then we'll do a hands-on workshop in RNA-seq. And then we'll start on genomics. In January, we will have a hands-on bioinformatics workshop, etc. So you can really look through the planned activities over here. You also have a page called faculty, and that will show you all of the people that are involved that you can be in touch with. So you can see here all of the people that, you know, if you need to ask about something, you can ask them right here. Okay, and then we will also track progress, so you'll be able to see kind of what is everybody doing. Okay, so back to the platform. The second area that is more practical is going to be right here. And what we will cover are specific sections in this areas of analysis. So we will focus on the next generation sequencing data, and we will cover a lot of that. So as you saw in the curriculum section, we will talk about transcriptomics. We will talk about single cell transcriptomics as well. We'll talk about epigenetics and genomics. So we'll talk about mutation variant, copy number variation. We'll talk about ChIP-seq. We will talk about bisulfite DNA methylation. And then we'll also talk about metagenomics and some non-coding RNA as well. So whenever we will do these different types of data, we will, go through kind of the background, and then you will be given a data set, which is going to be a link. So I'll just demonstrate what that would mean. So for example, the first section that we will do is going to be transcriptomics. And each section is built in a very similar way. So you have the top section that is an input. What is the type of data that you're uploading? And so typically you would use a reference genome with annotation with a GTF file. We'll talk more about it when we start the transcriptomics module. And here you have parent or single end. The type of input file that you will have typically will be a FASTQ file, and then a reference organism. And we'll try and use human genome as the reference genome. Then you will be able to upload a data. Now, I mentioned that for educational users, there's not going to be, there's actually a limit of 50 megabytes, so you can't really upload FASTQ files. But you can use this section called SVL, which is just a reference link for files that are already uploaded. And these are called projects, right? So you can either use multiple SVL links like this, or you can just upload a number of SVL links together through URL bulk upload. So for example, you can go to you can just upload a file, and then as you press on this, you will see that a lot of FASTQ files have been already input into the system, and you can drag and drop them into several groups. For example, if you want to do differential gene expression or something like that, or you can just cancel no groups. And now you can build your pipeline. So again, like I said, all of these methods, a lot of these methods are public domain methods. So if you search on Google for what is, for example, HTSeq, or what is HiSat2, or what is Bowtie, you can do that. But let's just quickly do that. Bowtie. Okay, and you can see right away that you can find the source code and you can see what it is. Now, also in the process of doing that, you will actually get help in the system, and you'll be able to find all of those things. So you can see that now I have a limited number of options available for me. Those that are darker are the ones that I can select. So I can, for example, build my pipeline reading about what methods I am using. And more explanation about the methods and the ideas behind them are going to be given in the online modules. Now you will have a reference publication that you can read, and a lot of times that reference publication will also give you the source code if you want to implement that in your own pipeline. So now I've connected start and top hat 2. And for example, I can use the standard cufflinks pipeline. So I can do cufflinks, cuff merge, and cuff diff. And you saw that each pop-up tells me what it is that I'm doing. But in the workshop, we will kind of look at how to set up the pipeline, what would be the input, what would be the output, and then how do we interpret the results. And so the focus of the program isn't going to be on necessarily the code itself. It's going to be on what is the logic behind this. And so as you can see, each number of methods is organized in a group, and you can also read about what that group does. So you can see here the different titles of the groups, and now you can click on them and read about what they do. So this is the platform, and you will see that there's going to be, so once I created that pipeline, you can see that it appeared right here, and I can monitor the status. So since I didn't click on start, it's only prepared, but I can always go back, and that way I can remember what exactly I was doing, and I can also run my pipeline. Now the completed pipelines, let me see something that is relevant here. So the completed ones will also have output files, and we will talk about what is in those output files and how do we use them. Okay, so why is this important? Well, it's important because if you go to Twitter and you type in bioinformatics headache, you will see, what really happens in real life when you kind of read. So this is the typical process. You read about some newly published tool that does this amazing thing. You take some time, you find the code finally, you run it, test it on a small data set, nothing works, and you forget about it, right? And so the goal of the platform is to eliminate this problem. We do the curation of the tools. We go and make sure that they work as they are described. We try and work through them with example projects, and then we prepare those as educational data sets. And then hopefully, instead of getting discouraged by other people doing amazing things, but I can't, you can actually see that you also can. And then you can take the things that are useful to your own research and implement them in your own way, however is useful to you. So how are these modules organized? So the way that they are organized is that we will have orientation workshops just like today, where we're not really doing anything useful, but we're just talking about what we will be doing. But we will kind of give it an outline. And so this is the first module. There's going to be introduction to bioinformatics. Now, each one is about a week long. So introduction to bioinformatics is really to test that we understand each other. There's not going to be a lot of useful information for you, I guess. It's mostly going to be about what is bioinformatics in general and how is it used in different scenarios. But it's a very theoretical, superficial overview. It's not going to be very in-depth. And it's just to understand what are the different components. So here I go for the online modules, here I go for the practical things, and this is what I'm going to learn. But really what we're going to try and do in the first month is cover transcriptomics. So how do we take next generation sequencing files from Illumina machines, raw files, and how do we translate them into tables of expression? And then what can we do with those thousands and thousands of genes? And so those consist of 3 modules, Transcriptomics 1, 2, and 3. They talk about processing data. Then they talk about standard statistical methods like t-test and what is differential gene expression. So we'll cover all the things that we all know about already, but in the context of RNA-seq, what are p-values, false discovery rate, all of those things. And we'll touch a little bit on regression. And then we will talk about other methods. How do we do dimensionality reduction? How do we do PCA, clustering, different things that can help us mine that data and extract something useful that then we can interpret? And how do we do then annotation? How do we convert the ensemble IDs to gene symbols? How do we do pathway analysis, gene set enrichment analysis, etc.? So we hope to do that in a month. And if you're already familiar with processing, how to run your cufflinks pipeline and what is the table of expression, you can just skip on to the next one because we will provide the data sets within these modules. At the end of this process, we will have a workshop that is intended to help everybody do the things that we already did here, but together in a session and address any kind of questions or challenges that come up. And then at the end, it's optional, but you are welcome to take one of the data sets and really work through it. So the idea is that you learn kind of bits and pieces, but if you can put the bits and pieces together on your own, that means you actually know what you're doing. And we will provide you with just the data set and some direction on what you can start doing with it, but then it will be up to you on how you complete it. Yeah, go ahead.
The analysis different for the data set generated from an Illumina system or an ion proton, ion torrent? What's the difference? But because you said Illumina.
Mm-hmm. Make sure you repeat your question because we only have a lavalier mic.
So to generalize the question, it was whether different platforms to generate data have an impact on how you analyze them. So we will talk a little bit about different platforms. We're not going to cover all of them, but the bottom line is that if you have a FASTQ file at the end, you can process the FASTQ file with the same tools. You just have to understand a little bit about how it was generated to be aware of what would be the specifics. For example, the length of reads would be something specific to a platform. Quality, coverage, things like that. So we will talk about those things. Any other questions? Any other comments? Okay, so and also online. So if anybody has any questions or comments online, you can probably put them in chat, and we will try to address them. To help you evaluate your understanding and your ability to kind of actually use this in the future, what we've come up with as a way to assess everybody's level and progress is to use the methods section of publications. So instead of just tests on do you know what this term is or that term is, we'll actually take the methods section and the figures from that section, and we will ask questions whether you understand what that means. And we hope to achieve two things by doing that. First of all, because a lot of people just skip that section, right? So we're interested about what they did, what they set out to do, and what was the significance and what they found. But how exactly, that's where it becomes technical and dry, and, not everybody wants to read through that. But it matters because your choice of methods of how you, for example, normalize the data, or how you, chose one method over the other, is it because somebody told you, or because you tried many different things and you chose one, will really play a role in the quality of the final results. And some things that could have been done differently to find alternative results that are interesting, or reveal something technical about the data. So we will try and go through at least several papers, so probably 2 papers in each module, that will talk about some of the standard processing, analysis, interpretation steps, and about different concepts. And these concepts are going to have biomedical significance. So again, for those of you that have, for example, plant biology background or interest, this is not to say that, that is less important. It's just that the focus of the program is a little bit more biomedical. So this is going to be both in the pre-assessment and the post-assessment. They're not going to be exactly the same paper, but the idea would be that, in the beginning, you will see how it's a little bit more challenging to exactly understand what happened in the methods. And then towards the end, you will start noticing a little more technical differences. Some people used k-means clustering and others relied on hierarchical clustering. And now I understand what's the difference and why they chose this and not the other. So that would be, I think, an interesting experience then. Okay, any other questions about the program or about the different parts of online and kind of in-person? Sahil and John, anything from the online community?
Still working with the credentials.
Okay. Okay, great.
He's monitoring as well.
Okay, great. Yeah? Did you have a question? No?
So the one comment we've got seems to be pretty straightforward.
All right, great. So, introduction to bioinformatics. So what is bioinformatics? Bioinformatics is an interdisciplinary field, and we talked about this already, but it really combines 3 major areas, right? We have the domain, which is biology, and then we have other sciences that are fields in their own right, statistics, computer science, and these are some other fields that have an impact on bioinformatics. The second part of bioinformatics is that it really started as a field of its own in about the 1960s. And so to get a perspective on why that happened, I put together this little timeline on how we got here. So in the 1950s, the DNA structure was discovered. And once that structure was discovered, right away, people started thinking, well, how do we assemble the human genome? So between the 1950s and 1990 to about 2003, there was a rapid generation of data from the genome, from the DNA. It was a big breakthrough, obviously, and today those 2 people continue to kind of play a role. So one of them is the director of NIH, the other one is Craig Venter. But then really what happened was that Illumina brought this power to generate this kind of data to the masses, right? Making it affordable and making it possible outside of these, large government-funded projects. And that was in 2006. So in 2015, we already had 1,000 human genomes assembled, That was again published in 2015, that it was entering the final phase, the 1,000 human genomes, the 1,000 genome project. And then today, in 2018, we have even more of that data. We have thousands of genomes. Okay. Oh, we're not going to go through all of that. But the idea is that we have a lot of this data that is now available. And as that was happening, so that was the story of the human genome, but really as that was happening, we discovered that the relationship between gene and phenotype is not as straightforward. And it actually is quite complex. And so as a result, a lot of other types of omics have started playing an emerging role in bioinformatics. And so bioinformatics as a field really studies these different types of omics data and what to do, how to bridge the information that we have with the data that has still not become information. It's still data. So we have phenomics, which is the study of phenotypes. For example, cellular images. We have the study of genomics. So this is an example of a VCF file. So you can see that the chromosomes and the DNA really are represented by this data. That what is it? Well, it's really how individual nucleotides are changed and their quality, and whether we can call those somatic mutations or germline mutations or other things. Interestingly, in about 2009, there was an increased interest in epigenetics. So this is the number of publications mentioning epigenetic data. So you can see that it happened not so long ago. But today we understand the critical role epigenetic modifications play in different processes. And those include histone modifications as well as different non-coding RNAs that modify how proteins get produced. And transcriptomics, where we study alternative splicing, the production of mRNAs and other types of RNAs, so the whole transcriptome. So the field is expanding by different types of data sets. And obviously, there's an interest in integrating those, because even though we think we understand each step of the process, really we don't know a lot about the missing pieces. And so the idea is that if we can know something about one part of it, and another part is known from a different types of data, if we integrate, if we know how they are related, we can then fill in the gaps. And so integration starts playing a critical role. And so today what you will see is a lot of multi-omics data sets that combine these different types of technologies to really understand both mechanistically what's going on and to try to interpret by leveraging what we know about one data type or the other, what this really means. The other part of bioinformatics is really driven by these different technologies that produce the data, right? So we talked about this, and we talked about the role of Illumina and other types of sequencers that made this data available. But these data sets are specific to the technology that they utilize. And so understanding the technology, understanding the limitations of that technology and the specifics of that technology also is very important, because those result in different types of data sets that require processing. So as you can see, these are FASTA files, which are just sequences. And now you have FASTQ format that also includes the quality of those sequences, but it's essentially the same thing. And so as you can see, to really understand something about it, right? So previously, like when the human genome was published, it was also printed in a series of books, and that's just unreasonable to process and analyze. And so people have come up with different types of processing pipelines, like we saw in RNA-seq, to take that raw data and translate it into something that we can analyze. And so as a result, what you will see on the platform are these different sections that deal with the machines that produce the data and transferring those into a readable table format. And so, we mentioned other high throughput sequencing technologies, but also microarrays, ChIP-seq, and then RNA editing. So either different types of data that we can extract from one type of library prep or another in the lab, or different types of techniques to actually generate that data in the machine. Now, another challenge that comes as a result of this type of data is really the challenge of data mining. So this is an example of a low throughput data set. A low throughput data set is simple in the sense that you have only a couple of variables. For example, here you have weight and size plotted in a two-dimensional plot. And here you can really see the relationships between these variables clearly. In omics data sets, those relationships are really non-trivial. It's really difficult to understand how are they related to each other and what can this all mean in the context of biology that is also not very intuitive to us, right? So here we kind of understand how size and weight should be related. And it's easy for us, it's intuitive for us to interpret. Here, it takes a lot of domain-specific knowledge and extracting that knowledge from publications, databases, etc. And so bioinformatics deals a lot with that. And so you can see how a lot of that will come into play, especially when we talk about high throughput data. So high throughput data means that for every sample, you have a lot of indicators. And so you will have, for example, tens of thousands of genes per sample, or mutations per sample, or other factors. Going back to the history, at the same time, around 2000, so when the Human Genome Project was published, you can see an emergence of publications related to machine learning. And that is another development that has really advanced bioinformatics, but also has made bioinformatics technically more challenging for non-computer science people and non-math and statistics people, is to understand how do these methods really help us? What do they do? Because a lot of them, transform the data in such a way that is then difficult to interpret back in the biological terminology. Or, how do I interpret, for example, dimension reduction when I'm talking about genes and expression and, nucleotides and things like that. So another challenge here is to understand the analysis methods and to connect them back to biology. So because of that, throughout the course, we will also make this other course available that you don't actually get an introduction, and it's not going to be its own module in the sense that I showed in the beginning, but it's going to be an ongoing thing that you can reference, and it will have different explanations of these methods, these machine learning methods. So they're going to cover things like, unsupervised analysis methods, like clustering, dimensionality reduction. They will talk about classification and other supervised analysis methods, network analysis, and others in the context of practical examples with biomedical data sets. So how do we use this these high throughput technologies to classify different subtypes of cancer? How do we understand associations represented by network analysis and things like that? So we hope that this is going to be an additional resource that you can rely on to understand better the methods. Okay, so do you have any more questions so far about any of the things that were mentioned up until now? Okay, so let me show you how these things are organized again over here. Okay, so this is again just a practical overview. And the goal of the first Introduction to Bioinformatics module is to really get all of us on the same page. So if I go to courses right here, you can see that the upcoming courses are going to be listed right here. So I can go to Introduction to Bioinformatics. I guess I should log out. Okay, so Introduction to Bioinformatics is going to start off with a survey where you can help us understand your background a little bit better. Please fill it out. It would help us adapt our future materials to your level. And then we will talk about some of the things that I already mentioned today. So, Introduction to Bioinformatics as a field, data-driven discovery. How do we go from hypothesis-based to hypothesis-free kind of data mining approach to all of these big data sets? There will be a practical kind of hands-on overview of the platform and what you can do with it and why it's useful and what scenarios. And then we'll talk about some of the biomedical applications of bioinformatics. How is this data analysis significant or impactful for different challenges and projects in healthcare and in other fields? And then there's a quiz. So all of this is really, you can probably complete the whole thing in 30 minutes, but it would be great for us to see, what you think about the length, the time commitment, the level of the materials. And we envision that for some of you, it will be really simple and probably not very informative. But for other people that come from different other backgrounds, it might be a useful resource to kind of get everybody on the same level. So after you complete this, so just practically how to do this, you have to enroll in the course. And then once you are enrolled, you can go into a lesson. Let me just show what that looks like. So you will have your lessons here on the side. This is the survey. After you are done with the survey and you have submitted it, here at the bottom, you press on complete. And after you have completed, you can then continue on to the next one. So all of this information is going to be useful for us also to kind of see where everybody is at so that we're not running ahead. And you see that now it's green, so I've completed it, and now I can go to the next one. So just remember to press on that complete button. And then at the very end, once you have gone through the whole course, the whole module, there will be a button to complete the whole module. So please click on that as well. And then the next course that will come right after that is going to be Transcriptomics. So I'll just, some of you might want to skip Introduction to Bioinformatics if you're already familiar with that. But here's what we will do in the first module of Transcriptomics. We will talk about the biology of transcription and some other methods to quantify RNA. And then we will jump into the hands-on sessions, which will include how to generate the transcriptome expression data. So what are some technologies that actually do this and how do we understand them? And then how do we process them? Some different examples of pipelines. And then what can you do with that expression table? So we will give you a table and we will look at different genes, expression values, etc. So these are the very simple first modules that everybody can start with, and hopefully that by next time we will have this meeting, everybody will have all of their technical details figured out and all of their accounts working. Okay, so this is what I had for the introduction to bioinformatics. And I'm happy to answer any questions that you might have, or if you have any other specific things that you would like to know about anything that I've shown so far, I'm happy to go deeper into that.
So there's a question on the chat from Melissa that has the introduction to bioinformatics course been updated or anything since this summer?
So some of the materials have been updated. I do have to say that we update regularly the platform itself. So Introduction to Bioinformatics has been only updated a little bit, but there's nothing new for people that took it during the summer. There are no substantial changes. On the other sections, we have been making updates to the platform and to the modules themselves. But as a general rule, I think that people that have completed the summer course would probably benefit a lot more from going on to the other modules that we haven't touched in the summer. Yeah.
Maybe I missed it, but would you recommend doing these online modules before each practical session, or just briefly glance at them before each?
Yeah, it would be best if you could try and do them before, because what we found was that during the session, we want to achieve the practical results during the duration of the session. We only have 2 hours. And so it's good that you already know what you're doing a little bit, and we can focus on kind of not the very basic technical challenges, but the more conceptual questions about why this and why not that, and how do these, relate to each other, and really try to avoid, how do I open a file in Excel, or, where do I find my files, or how do I connect two methods together? Those types of questions are all covered in the online modules. I understand that they could be a little bit more dry, just having to do it in your own time. But I think they're fairly quick. So if everybody can try and go through some of the practical things online before coming to the session, it would be great. And we actually can see the progress. I can, I think, show you a little bit of what that would look like. But basically, we see on our end who is doing what, at least in terms of progress of these online courses and your pipelines that you are taking. And so that helps us really in 2 ways. One is to see whether specific sections are more difficult or take more time, for example, or people are having more technical issues with those, and then we can address that in more detail and help everybody during those in-person workshops. And the other way is to put together groups when we do independent projects. So in an independent project, obviously, you want to put together a group of people that can help each other and that can make the experience more smooth. And so we try to match people that have a little bit more technical experience that we can kind of see through analysis of the data that we get, and the people that might have more insight into the biology and the data set so that they can kind of inform each other. Yeah.
It's all done RStudio platform, actual analysis, the pipeline, workflow.
So these are, the platform itself takes executables that are written in different languages and runs them through a series of bash scripts. So really what it is you can have, in script in R or in Python or in C or whatever it is, and then the bash scripts will just execute them in the right folders with the right reference files and libraries, and etc. So the platform will help dealing with the big data sets and getting them to a level that we can look at them. How we look at them, we can look at them in Excel, we can look at them in Notepad, we can print them out, we can use RStudio. It depends on what's more comfortable for you. We will do a little introduction into some methods in R, just because some of the machine learning methods are like a standard 2-line code in R. And so it's easy to run it and look at the output and the input. But we're not going to be going into detail. And I'm sure that there are other more detailed R courses that people can take. Or if you have any specific questions, for example, Lyndon, I'm sure other people here could help.
I have only a pre-version or basic version of RStudio, and that should do.
Yeah, you actually do not have to have R. We'll just kind of use it as a general example. This tool exists, and this is what it does, and this is how you can create a heat map, for example. But we're not going to be going into syntax or how to write your own code. Yeah.
So while participants trying to practice in your module, and then if they have some question, and what's the best way to contact you or your team?
Yeah. So yes, if you have any technical difficulties, something's not working, or you don't know where something is, there's going to be a chat box right here. And this chat box is going to be consistent between the educational site and over here for the server site. So we will respond within 24 hours of posting that question there. It would be good if you can add your name and your email so that we can understand whether you're a part of the LBRN program or some other program. Or you can reach out to us by email. And then we will get back to you on that as well. So we will answer questions like, something's not working, I'm having technical difficulties on a specific thing. And if there are more general questions like what does this method do, or why this method and not that method, we will typically post an answer in the forum. So if you have those kinds of questions, because we want to make that available to everybody. I will just show that here. So under LBRN organization, which is right here, there's a forum. So you can post, if you have questions on anything, just post it right here. And then we can submit a detailed answer, and then everybody will have access to that. Okay, well, if that's all the questions, yeah, go ahead.
Which courses should we be looking at before the next session?
So we will start with transcriptomics. So introduction to bioinformatics and transcriptomics. You don't have to, complete any of them before the next session. The next session is going to be an introduction to transcriptomics. But if you do like transcriptomics 1, it would be helpful. So the next session is going to be, again, you can look at the schedule right here. What's that?
Right.
Yeah. So you can see that this is today's, or this is today's, this is the next one, introduction to RNA-seq. So if I click on this one, you can, if you're logged in, it will populate this automatically, but you can, register through here or register through the LBRN website, and then we will know that you are interested in this. Again, the point of this is that there will be multiple things happening at the same time. So we will start genomics right after that. So if you're more interested in mutation variants and copy number variation, sign up for genomics. you're more interested in gene expression, sign up for transcriptomics. And so we'll have them in parallel. And so that's why we're asking for people to register.
Where did you register at? What link are we on?
Register for the events.
Yeah.
So you can either go to this page, which is the curriculum page under the LBRN organization, and that will give you all the links in one single place. So for example, I clicked on this. So these black ones are the events, and these are going to be your modules. So if you click on this, that's how I got to that event page. But also we will post this link on the announcements on the main page, just so that everybody has that. And then just a direct link where you will see all of these once you're logged in is if you just go to events. The list of all the events, so if you want to sign up for future ones, you can see the list right here. Okay, so I think we're done with the presentation. We are going to be here for a little longer, so if you want to talk to myself or some of the people that are involved from the LVRN side, Sahil is also here. If you have any issues with your account, you don't know how to log in, or you don't have your credentials, we can solve that right here, right now. And of course, if you want to just talk to us and tell us if you're interested in doing an independent project as an overall part of this program. I think the goal is to have at least a couple of those independent projects. So over the course of the next couple of months, we'll be looking forward to hearing from some of you if you actually want to pursue that direction. Just let us or some of the administration here from CCT and LVRN know. Thank you.